I'm Justin. I'm a scientist at the Allen Institute, and I'm going to be talking to you today a little bit about what the Allen Institute does, some of the things that we're building, and how Python is a key aspect of our software architecture. Is this thing on? So the Allen Institute for Brain Science, we've been around for a few years. We, the larger organization, I've not been here for its entirety. And the Allen Institute is kind of interesting and unique in terms of research institutes. So we have a very strong commitment to open data, and we take a big science, team science approach to what we produce. And over the years, we have released multiple very large data sets that are of general value to the neuroscience community. And our most recent data set is the Allen Brain Observatory, which was released last May, and we've had 2 subsequent releases of it as well. And that's what I'm going to be talking a little bit about today. We have what is the point of the Allen Brain Observatory? So within the visual system, we have a sense of different brain regions which do different things. So the retina seems to maintain, the retina seems to maintain visual information, a somewhat veridical representation of the world. And as the neurons project up through the visual system, as we go to increasingly deep layers of the brain, the representations get more and more complex. And this architecture has inspired many neural network architectures that you guys might be familiar with, where an image of a cat eventually becomes represented as the concept of a cat. And a lot of this work has been done in primate models. But this model, this data, this architecture is insufficient to explain a lot of what we know about how the brain works. And there's kind of 2 key things that I want to highlight that this model is insufficient to characterize. One is that within each of these different colored layers, there's actually multiple different types of cells, and there's characteristic ways that those cells are connected to each other in order to form kind of local computational circuits that isn't really captured in these simple models. And secondarily, there's also a lot more of the brain here, and the way that these representations and the way that these different parts of the brain work may change based on the demands on an animal. So if you are, or a person. So if you are looking for a cat or trying to attend to a certain region, looking for a cat in a certain area, you might miss the cat that is somewhere else in your visual scene. If you are, so depending on your motivation, depending on what you're trying to do, the way that these different layers might work can change. So the Brain Observatory is the Allen Institute's attempt to basically survey the visual system, and particularly in the mouse visual cortex. And one of the reasons that we are looking in the mouse visual cortex is that we can identify, create transgenic animals who have cells in their brain that glow that glow when they are active, and we can specify certain cells to fluoresce when they are active. We can then record from multiple different visual areas. So this is an image of the surface of the mouse brain with different visual areas highlighted. And we can zoom in on, using a microscope, zoom in on a particular field of view, a particular region, and record these cells actually firing and actually active while we're presenting a bunch of images on the screen. And so based on all of these images, we can then look at characteristic ways that these cells respond, and this gives us information about how the visual system is working. And ultimately, all of this data is released online free for you to use. So if you go to observatory.brainmap.org, you can browse this data yourself right now. If you do pip install Allen SDK, then you have our entire data set of 40,000 neurons at your fingertips. I'm not going to be talking to you about all of the kind of ETL, the data pipeline that we have for this entire data set. But instead, I'm going to talk to you a little bit more about how we get there. So we have in order to generate these data sets, we have a number of teams that whose activities are coordinated, who have different roles in producing these data. So we have surgery teams who do the surgeries. We have our behavior team that helps train mice. We have our physiology team that does the actual recordings. And at the end of the day, one of our goals is to really understand this full cycle of how the brain works and the entire system that the brain works in. So what I described to you so far, mice were simply sitting in front of a screen with images being presented to them, and that captures kind of this environment to perception corner of this loop. But ultimately, when we're really talking about what the brain does in the world, the brain, our brains, we use that information to make decisions. We use that information to make decisions that we can then act on those decisions. When we act on those decisions, that changes the world around us. And so there's a full cycle that we'd like to capture. And so in order to do that, we actually need to train mice to do something while we are recording. And so we are building out a visual behavior brain observatory, in which we'll expand out a very key piece of this data production pipeline to be able to train mice in a couple of weeks to be able to make decisions about things they see, so that when we go to record from them, they're also making decisions about what they see, and we can capture that full cycle. So in order to do this, though, we need to train mice at scale. So how do we train mice at scale? Well, the key pieces is that we need to automate whatever we can. Whatever we can't automate, we need to be able to support our trainers. And then beyond that, we just need to be able to iterate and improve our ability to train animals. So the first piece of this, so the basic task that we're going to be presenting to animals and challenging them to do is to lick a water spout when they see an image change. So we're going to have multiple images that are presented on the screen, and when the image changes, they need to lick a water spout. And in order to train animals to do this, there's a fairly basic algorithm that's been formalized for many years and used for many years beyond that to reinforce their behavior. And basically, we follow this little graph. So did the image change? If the image changed, then we ask, did the mouse respond? If the mouse did respond, then they get a reward. If the mouse did not respond, then maybe they'll get a reward next time. On the other hand, if the image did not change and the mouse responds, then they need to be presented the trial again. And if they did not respond correctly, then they'll get another chance soon. Now, this basic logic has been used for many years. So here's some cats that have learned a similar task. So they've learned that when they ring the bell, then their owner gives them a little bit of food. And this owner has basically scaled this up effectively to 2 animals, but it's much harder to scale this strategy up to dozens of animals simultaneously, which is what we need to be able to do. Luckily, it's pretty easy to get to get computers to do some of this for us. So our key challenges that we face in scaling behavior is first that we need to be able to minimize the intervention of our trainers during training. We need to be able to let our trainers monitor the animal's performance during training. Our scientists need to be able to analyze the behavior, both for its scientific value, but also to be able to iterate and improve our training. Because ultimately we want to be able to minimize the training time so that we can fit this into this full production workflow. So I'm going to be walking through each of these and talking about how kind of the tools that we've built out into our software architecture to solve these challenges. So here's my overview slide, and I'm going to drill into these shortly, of our software architecture. So effectively down here at the bottom, we have kind of our file storage, files and databases. We have a service layer that helps to mediate our interactions with these. The big blue box up on the left there is our behavior control workstation, which is where the primary point of interaction with trainers. And then the other blue boxes are other containers, and I'm going to go through and talk about what all of these are. So first, the core our first challenge is to minimize trainer intervention. And so our basic solution here is to automate our training algorithm and to be able to support efficient trainer workflows. So this all, first and foremost, gets drilled down into this one yellow box, which is our actual task. Our task is just a Python script that runs on a computer. This basically implements this logic that I talked to you about before in a task flow. So there are different windows that when we say, okay, we're going to wait until a change might happen. We have some window in which a change might occur. If a change occurs, then we're going to have a window in which we listen for responses. So this all has been implemented in software, largely using PyQT to handle Qt events, so that this can all be asynchronous, and we can just trigger these windows off of each other. All of our hardware interactions, so actually monitoring the mouse's behavior and controlling the solenoid to present water, is using PyDAQmx, which controls National Instruments cards. All the images that are being presented on the screen is using the PsychoPy toolbox. And then this also uses some 0MQ in order to publish events live, which I'll be talking about shortly. We need to be able to scale this up, though, and here's one of our behavior training rooms. So each of these boxes is a training box. Those little syringes are where the water comes from. That's the reward for the animal. Off to the left and right are two of our control workstations that our trainers use to actually interact with the mice. So in order to scale this up, though, and down there along the very bottom are the actual computers that are running the software for them. So we need to be able to take our task and we need to be able to deploy it, because this is running on multiple different computers. So we basically have a task deployment GUI that the trainers can use to load up the task for an animal. This can actually read directly from our service layer to identify the proper task to give to the animal. This uses 0MQ to send that script and its associated parameters to the agent that's running on the computer, the mouse's computer. The agent then deploys the task, which runs. And then when the task is done, it saves itself to back onto disk so that we can look at the data later. Nicely, though, because we have every single control computer controls 6 individual computers, and so this gets deployed in parallel. We've got a nice little GUI that was all written in Qt in order to load this stuff up, and then to use 0MQ to send things along to deploy the actual tasks. We want to be able to make this even easier for our trainers, though. So this interface, whereas we can sit here and type things in if we want to minimize the errors for our trainers, we've actually made it even easier such that the trainers only have to scan a barcode on the mouse's cage. They scan a barcode on the training chamber, and then they just have to click Run in Script GUI, which is the name of our little task deployer, and it runs. So our next challenge is to be able to let our trainers monitor performance of the animals during training. So our solution to this is to stream our training data and to present our visualizations on a central dashboard. So here's our dashboard. So these are just web interfaces that present an overview for each trainer. They've got a tab they can click on. They get their overview of the data as the mouse is running. And this gets regularly updated with the most recent data from a given mouse, and then we can drill in down to get more specific plots from individual mice as they're running as well. And so this is all basically built on top of Tornado, presenting the web service, again using pyzmq to handle 0MQ messaging, and then using VEGA to define our plots. And so each of our computers that the tasks are actually running on are then publishing regularly, which goes off to an accumulator that pools all of the data together and then bounces it back off to our monitoring dashboard, which presents the web interface that our trainers can then use. So our other challenge is that our scientists need to actually be able to analyze this behavior as well, and analyze the animal's behavior. So, unsurprisingly, perhaps we've built out a shared analysis and visualization package that we use internally in order to load up this data and analyze it. So this package, basically, we have a bunch of standard plots that we use that we can analyze after every single behavioral session. We can analyze that animal's data, get some summary information. On the left here is one of our plots for a single session that our scientists use in order to analyze the animal's behavior. On our right is a summary of that animal over multiple days. And so we can generate these plots every night so that our scientists can get a feel for how a given animal is doing and how it's progressing through its training. And this is all built on top of NumPy, SciPy, matplotlib, pandas, seaborn, kind of the standard scientific analysis stack. This also, since we've built this all kind of on top of pandas, in addition to doing kind of these standard reports that we push out, we can also drill down into more in-depth analysis. So we've got Jupyter notebooks, and we can drill down into more specific analyses. So here's some analysis that I did a couple months ago, looking at the sensitivity to a bunch of different mice along the x-axis there, to the timing of when the changes occurred. So this was just to see whether or not animals were sensitive to when these changes were happening, and whether that was affecting their behavior. And indeed, it seems to for most animals. We're able to do a lot of fun things with this. So this is one of our, we've been exploring training animals to discriminate between digits in the MNIST data set. So we can flash multiple zeros within the MNIST data set, and then at some point it changes to a 1, and the animal needs to lick if the category changed, but not if the individual MNIST exemplar changed. And so what this is plotting is the likelihood of an animal licking for any given combination and any given transition between the digits. So the dark purple boxes indicate that the animals are less likely to lick within the category changes, but more likely to lick when the categories do change. So effectively, our behavior analysis package kind of sits over here, that it's largely kind of reading out from this from our data stores in order to analyze our data. So one of our final challenges is that we need to be able to automatically progress mice through these training stages. So when we have a lot of mice going simultaneously, we can get in order to train these mice to do something like the MNIST data set, we can't start them on something fairly complex on day 1. Instead, we need to start them on a much easier version of the task, and then move them on to harder versions of a task. So what we've done is we've automated our analysis of past behavior in order to determine future training parameters. And so effectively, what we can define is we can define a graph. We can define a graph of stages of training that in the simplest version is linear and goes from easy to hard. So every node in this graph is a training stage with a set of parameters that are associated with it. And then each transition can occur that an animal can advance to the next training stage if some performance criteria is met. So every day we analyze the we need to analyze their behavior. If they're doing well enough on the task, then they can get tripped on and go to the next stage in the task. This is all built using a combination of ZeroMQ in order to monitor when animals are done with the task. But all of the state machine logic is done using Python package called transitions, which is fantastic for defining these kinds of state machines. And so this automatic training algorithm basically sits here and monitors the accumulator in order to get updates for when the animal's done with a training session. When the animal's done with a training session, that triggers an analysis from our automatic training algorithm that uses the packages, our analysis packages that we've developed to analyze their behavior, and then makes a decision on whether the parameters need to change or not. And if the parameters do need to change, then that gets saved back down into our data store, which then on the next day can get loaded back up automatically by our trainers when they scan the barcode. So kind of as an overview for some of the, I think one of the things that's remarkable about this is that this isn't just one little corner of analysis that's using Python, but our entire architecture is built using Python at every level. Everything from dealing with distributed messaging down to hardware control, all the way up to building out our web visualizations and building out our scientific analysis. And so this is all. This just kind of, I think, highlights a lot of the points that Jake was making earlier about the power of Python in the data science sphere as being not just about data science, but about this single tool that has so many other components to it that can all work together. So using this, basically, over the past few months, we've been trying to iterate and improve our training in order to get our animals trained up fast enough to be able to work within our pipeline. So here's a summary plot that I put together. So here's the number of training days. Each color here is a different cohort of animals that all had similar training parameters, going from the darkest being our oldest cohort to the lightest being our most recent cohorts. And so over this time, we've basically been able to increase our throughput and decrease our training time, which ultimately will get us ready to be able to deploy this into our full data generation pipeline, and hopefully around, well, I'm not going to make any promises on time, but at some point in the not too distant future, I hope, we'll be able to deploy all of this data will get deployed to, along with all of the Allen Institute's other data products, to observatory.brainmap.org, and you'll be able to download all of it with a simple pip install. So I'd like to thank especially all the people who worked on the different components of this. So the neural coding group, our manufacturing and process engineering group, our modeling analysis and theory group, as well as our neural surgery and behavior group who trained up all the mice. I know that we've got jobs open for software engineers and for scientists. So if you're interested in the kind of things we do, you should check out the website. And more than anything, I'd like to thank Paul Allen and Jody Allen for their immense and continued support. Thank you very much.
Thanks. Before I go to questions, there's a quick announcement I have to make. So if you're in this room, you're in the correct room for the pub quiz. So the pub quiz that's coming up after the talk is going to be held here. However, you can't have the pub quiz if you don't have, if you're in a pub, you have to drink something. So there's going to be drinks outside. And so I don't know how long the questions are going to take. We do have about 20 minutes, so probably there'll be time. But if not, just so you know, if you want to have a drink at the quiz, you should step out at some point during the questions to get a drink and come back so that we can start on time. Okay? Okay, let's open up the floor for questions.
what are the range of things that the Allen SDK can do?
That's a good question. Let's see. What's the Allen SDK? What do we have in there? I've got some colleagues out there who can help me answer these questions too. I think Nick might be better able to.
Most of the recent web products, the mouse connectivity atlas, thank you. So the Allen SDK has access to 3 major data resources: the cell types database, or we call it the IVSCC, the in vitro single cell characterization pipeline, which is a comprehensive study of the intracellular voltage of, I don't know how many thousands of cells, but thousands of cells. There's the Brain Observatory, which is 2-photon imaging. Looking at the cortical responses of populations of neurons to passive viewing of visual stimuli. And then what's currently pulled up on my laptop that I was just browsing through is the Mouse Connectivity Atlas, which is a comprehensive track tracing in 3 dimensions of the axonal projections of different cell types throughout the mouse brain. So accessing the raw data, pre-processed preliminary results, and also algorithms that were used in the processing pipeline. Those are the things.
Anyone else? Actually, I have a question. The slide that you showed at the very end with the progress in training time.
Yeah.
So kind of double question, what were some of the things that you changed to improve that training time? And secondly, was it just trial and error, or do you have some more sophisticated ways of figuring out the kind of features that improve training time?
Yeah, I mean, it's one of these things that let's see. We have, I mean, there's a long history of some of this behavioral psychology work. And so, I mean, ultimately we're talking behavioral psychology when we talk about training the animals. And so part of it is that we have, there are kind of known tricks that it's kind of wondering whether we need to pull these things together into a grab bag. So a little bit of it is trial and error. So for example, there's questions about the timing of the distributions, or whether or not if the animal is in a timeout period, whether you give them feedback on that. So for some of those questions, we basically, if we had a candidate intervention, we did some within cohort splits, so sort of like an A/B test to be able to do explicit comparisons. Ultimately, some of the key pieces that facilitated the timing speed this training speed, basically had to do with the timing of the distribution. So when were these changes likely to occur, and to prevent the animals from guessing that too much. So forcing basically an exponential distribution, sampling from an exponential distribution instead of a uniform distribution. And then the other one is basically making the early stimuli as easy as possible. So that training time that I plotted there, it's a little bit, so that was basically the amount of time to get out of stage 1. So that was the first day that we knew that the animals were actually paying attention to the screen, that they were doing something where they were looking at, they were making decisions based on what they saw on the screen. So basically, by changing from a kind of hard to see stimulus to a very salient, big white and black lines on the screen, was one of the major changes that helped support training times. And then pairing that with the automated training algorithms and implementing the automated training algorithms, that as soon as we get a sense of an animal that's performing well, then the training algorithm can kick them on to the next stage, instead of a scientist getting distracted, going off to a conference for 3 days, and forgetting to check whether or not they should advance them along.
Yes.
My question is, have you seen any transmission of memory or studies on generations of mice who were trained, their offspring are
faster at going through this versus a generation where they've never seen anything like this, and you know they're coming in from the wild?
Yeah.
Any differences in training time for those types of things?
That's a good question. I don't know specifically. So these mice have been bred particularly for these transgenic indicators. So they are so they're bred so that they have this protein that fluoresces in very specific cells. So all of our breeding strategies are centered around those issues. I think it's a fascinating question, and it gets to it gets into very complicated questions, I think, in the field of neuroscience in general about some of these like nature versus nurture questions, and what aspects of behavior are endogenous, what do you come along with when you're born, can you learn along the way. I don't know off the top of my head of anything that has really looked at that explicitly in this context.
Yes.
So I thought it was kind of a big jump to go from image changes to image recognition. Was there a stage in between it all where you looked into something more simple, like the intensity of light and the color of light, before actually reaching images?
Yes. Yeah. So So you mean in the context of the training itself, or?
Yeah, and just like, I guess I was thinking, are these mice actually seeing images, or are they just simply reacting to the environment?
Totally, absolutely, yeah. Yes, so basically, that's one of the key questions that we need to like ask and validate within a data set. So for example, how do I go back here? Sorry. There we go, okay. So like for this, I think this MNIST data is a good example. So to what extent is this mouse, so this mouse is able to, for the most part, doesn't respond when zeros go to zeros, but does respond when zeros go to other digits. Is that because they have extracted the higher level features of these images so that they have a sense of zeroness and oneness, or is it that basically there's light in the center of the screen or not? And that's basically an active area of inquiry. So in order for me to make a strong claim here that this mouse is doing some categorical discrimination or has some categorical perception, I'm not willing to make that claim right now. But I think that it's very interesting, though, to be able to pair that with the types of neural data that we can get, because what you might expect is that basically we have a sense of different parts of the brain being involved in these different types of decisions in different ways. And so ultimately, whatever we train animals up on and deploy to be able to download, like, you yourself would be able to go and download that data and say, okay, like, is this area responding to these images as categories, or is it responding to these images as some low-level feature that can better explain what this cell is doing? So.
Okay, anyone else? Okay, let's thank the speaker one more time.
Great. Thank you.